Okay, good afternoon, everybody. So I will be talking about beta. So I have several parts in my presentation. The largest one will be about B-cell epitope prediction tools, and then I will tell about homology modeling tools, and then a little bit about epitope viewers, how to use it, and then about the tool which called PIG, which we serve on our website but developed by others. So there will be 4 different tools discussed in my presentation. No, more tools, but 4 different parts of presentation. B-cell epitope prediction tool. Yeah, I put that slide for protein data bank, so to show you where we take data on structure. So from protein data bank, we take data on complexes of peptides with MHC, antibodies with proteins, and other antigens. So, and those all data which are in PDB are presented in our database. And actually also PDB structure serves as a basis for developing structure-based tools in our database. And in homology modeling tools, which I will show, we also use structures from PDB. So when we talk about structural epitope, you can think about that as a part of protein. In that case, it will be protein in white, which is recognized by antibody. And antibody binding site is also called as paracol. So, and we can distinguish between linear and discontinuous epitopes. And any protein can have both linear and discontinuous epitopes curated in our database. And for example, in this case, for myoglobin, you can see that linear epitopes are usually just stretches of amino acids on the protein. And the discontinuous epitope is also called conformational or non-sequential epitope, are just residues which grow together on the space to be recognized by antibodies. So, and structural methods such as X-ray crystallography, nuclear magnetic resonance, electron microscopy, they define so-called structural epitopes, how we call it. And other kind of methods, such as mass spectrometry, surface plasmon resonance, and different immunoassays and alanine scan, they are so-called functional methods. They define functional epitopes. And why it's important to differentiate between structural and functional epitope, I can demonstrate on this example. Here you can see in blue, red, and cyan are colored residues which are part of structural epitopes. So those residues which we know from the structure of antibody with complexes with protein, which I interact with antibody. And one residue here, which is in magenta, is not part of structural epitope, but it's part of functional epitope. And it's curated in our database as functional epitope, because we know that substitution of that epitope in some experiments markedly reduce antibody binding activity. At the same time, some epitopes, like red here, I like it in red, they can be both part of structural and functional epitopes. And some residues can be structural epitopes, belong to structural epitopes, but for example, we don't have experiments where they are shown to be functional, just because probably no one did such experiments. Okay, so that is how we can distinguish between functional and structural epitopes. And I will tell you later on why it's so important. So if you look at the history of B-cell epitope prediction, so the first method appeared in 1981, and then, and it was based on amino acid scale. Then later on, when more structures in PDB became available, first methods based on 3D protein structure appeared. It was like 20 years ago. And then in 2 years, the first structure of antibody protein complex, in that case with lysozyme, was solved. Only in 2000, the first database collecting epitopes appeared, which is called SIM. So in 2005, we released IEDB, and then more and more structures accumulated in PDB. And in 2009, there were more than 260 structures of antibody protein complexes, and that allowed us to start making structure-based methods for epitope prediction. And such methods as DiscoTope, ElliPro, BepiPred, and others were published in 2009 and later on. So I will talk first about prediction of linear epitopes and then about conformational or discontinuous epitopes. So there are two groups of methods for prediction of linear epitopes. The first group is based on amino acid scales, and the second is based on machine learning approaches. So the first method was published in 1981, and just like for historical reasons, I probably should mention it was based on calculation of residue hydrophilicity and on the assumption that hydrophilic regions in the protein are predominantly located on the surface and then accessible for antibodies. And they used 12 proteins with immunochemical data existed to check the predictive power of that model. And what's interesting that some predictions were later confirmed by experiments. For example, this one. Later, it's very recent work. So it's still interesting. So it's very old method, and it's available. It was published as a code in a Fortran language, by the way, in the paper. So, and in the database, you can find methods for linear epitope predictions under the detailed epitope prediction tool. And if you click here, we can select some examples, for example, hepatitis A virus. And again, it will be cut and paste here sequence, or you can provide a Swiss protein ID, for example. And then there is a number of methods which you can choose from. So, and some of the methods, most of them, except BepiPred, which is a machine learning method, other methods, they use amino acid scale. For example, let's look at Parker hydrophilicity prediction. So when we submit, that's the result you would see. You would see the graph for score predicted. So on the y-axis, there will be hydrophilicity score, and on the x-axis there will be sequence position in the sequence submitted to the method. And depending on the threshold, which one can change here, so that will be our threshold by default. Just by eye, you can say, okay, those regions have very high hydrophilicity, those very low. So, and then it's calculated based on window size. So the window size can be changed. For example, in that case, it was 7 amino acids. And if it changed to 1, you will get something like that. So that's how you can play with the tool. And also change the threshold, and you can be more strict in your predictions. And also the values or predicted values can be output in table format. For example, here you can see that for each position, for each residue, there is a score calculated based on the selected window. So we had a window of size 7. So in the middle, you would see your amino acids. And so for that window, you would get that score. And it can be sorted. Yes?
Yeah. How do you choose your threshold? How does it work? Start with this graph.
So the question was how to choose the score and decide if it's a good prediction or bad prediction. So we don't have any recommendations here because we never actually benchmarked that. So and what we use as a default, that's what recommended by the authors of the method. So and those methods we just implemented on our website and we put them on our users, but we don't kind of stand for what they actually do and how they perform. Yes.
So, what the reason is, it doesn't optimum length, just like for
Okay, the question was if there is any recommendation of how to select the window size, is any recommended length for the size of B-cell linear epitope. So we know how the size ranges. So when we, from the database and from the structural data, we know that they range from like 5 amino acids up to 25 amino acids. So, and somewhere in the average would be 15. So probably it would be nice to play with the size between like 12 to maybe 16, something like that. But again, exactly how better to select the window size for any specific method, it was not done. So it can be done in the future, but in that case, we can provide specific recommendations. So in that case, we just recommend they use what author suggested. So they use by default 7. And again, so the data in the table can be downloaded in Excel files. So let's look at another method. For example, okay, so and so we have all those methods, and they kind of give the same output. For example, that would be Parker hydrophilicity prediction. That one would be based on any surface accessibility prediction for the same protein. And another one is based on Cho-Passman beta term prediction scale, and that's based on Carpus flexibility prediction. So they're all based on amino acid scale. So each amino acid from 20 standard amino acids assigns some value. And here is just those values are taken and averaged on the window size. So it's a very simple approach. And it probably, it's so simple, it shouldn't basically so in reality. But no, so that's what I'm trying to solve. So we cannot recommend anything in that specific case for using amino acid scales, but we can recommend to look at all of them. For example, when you look at all of them, you can start getting a consensus by eye on your own. For example, like this place, it was predicted by also method as epitopic region, the region where antibodies can bind. So, and probably if you're trying to use those methods to do experiments, it would be the first region to look at. So it's just so that kind of recommendation you can give. So, and now I need to tell you so how we measure the performance of different methods. For example, we have the protein. The protein will be here, like the whole stuff in white. For example, it has 156 residues. And this in red is our known epitope. So, and some methods, some XYZ methods predicted this area as the epitope in blue. So now we can calculate the number of correctly predicted epitope residues, true positives, false negative values, those which were predicted as negative, falsely not epitopes, false positives, which were predicted as epitopes, epitope residues, but they are not, and all the rest will be as true negative. So from those four values, we can calculate sensitivity, specificity, and accuracy. And then what we can do, we can draw the ROC curve, where those values as calculated from those numbers, the true positives, false positives, and negatives, can be on the x and the y axis. And when we change parameters of our method, for example, we change window size, or we change threshold, and for each changes of parameters, we can put a dot on this curve. And for example, for blue method. And then we can do the same for another method, which would be red. And then how we can compare those two methods? We just calculate area under ROC curve. And it can be sometimes calculated very simply, just from those numbers, sensitivity and specificity. So it will be a rough estimation, but in many cases it's a very good estimation. So, and for example, if that red method gave 0.64 and the blue method gave significantly higher value, we can tell that blue method performs significantly better than red method. So, and I will be using those ASC values and sensitivity when I will discuss down the method. Okay, so those definitions were used for evaluation of amino acid scale. So the first one was done in 1991, and 12 different scales applied to 85 linear epitopes. And they gave sensitivity of about 50–62%, depending on the scale. Then later on, the same group of authors, they showed that better strong scale can give accuracy of 70%. And then again, the same group and some other people, they try to combine different amino acid scales and see if it will improve the prediction. But their conclusion was that combining did not significantly improve the prediction. And then there was a paper where about 500 different amino acid scales were evaluated on 50 proteins. Using peptides from the antigen database. And the authors demonstrated that single scale amino acid propensity profiles cannot be used to predict epitope location reliably. So they showed that basically any scale was able to predict epitopes well, epitopes taken from the specific database. So, and then we did evaluation of methods, different methods, and the same methods which I just showed you, which are available on our website. And again, on data which we had at this time in IEDB, we showed that no method gave AUC value above 1. So they all performed not very well. Okay, so disappointed by all these scale approaches, people try to use machine learning approaches to predict linear epitopes. And the first method was published in 2006. It's called BepiPred, and it's available on our website. And the reported AUC value was 0.66. And among other methods, which we do not host on our website, I can tell BepiPred, and labs, and they gave different AAC values and different specificity and accuracy. And they use different approaches, support vector machine and artificial neural network. So, and if you are interested, you can look at those methods if they are available as web tools. They should be, but I'm not sure in that case. Okay, so let's look at BepiPred. Okay, BepiPred is available also here at the prediction of linear epitopes. And again, you can select hepatitis A virus as an example and select BepiPred. So we keep all those tools together because it's convenient to use, because they give the same output. But as I mentioned, they based on completely different approaches. So, and if those 4 methods are based on animated, 5 methods based on animated scales, this one is machine learning method. So again, you will see the familiar graph, and the threshold can be changed. So the threshold which is established by authors, which they recommend, and below predicted peptides, predicted epitopes. So that method gives not only scores, but it also predicts some peptides. Okay, and changing threshold, and you can also download the plotted values for each position in the table format. Okay, so and to conclude the discussion of linear B-cell epitopes, I can tell that more than 10 of different methods available as web tools, and no one gives AAC values above 0.75 and also accuracy above 75%. That means if you have 100 predictions, 100 epitopes, you know the epitopes, and you predict, you try to predict epitopes from the same protein, you would have only 75% overlap. So accuracy is 75%. And poor performance might be explained by their benchmark data set, which contain not genuine epitopes present in native proteins. And basically, it's a bit of hard problem in terms of benchmarking linear B-cell epitope prediction tools, because of making a good data set, good benchmarking data set. It's for everything. It's just like conclusion, like the whole conclusion. Yeah. Okay, so now I will change gears and talk about prediction discontinuous B-cell epitopes. And they all are based on 3D structure of proteins, or proteins and antibodies. And I will tell about which methods exist, and later on, Bjorn will tell about a new method which we recently introduced on our web tool, but I won't tell about that, so he will tell, okay? So that's not full picture what we have. So, and then we discuss how well do they perform. And so again, let me remind you about difference between structural and functional epitopes. So, and when we talk about structure-based methods, we use only structural epitopes. We do not use functional epitopes. And usage of functional epitopes is questionable because they might not correspond structural epitopes. And because of complexity of protein structure, you can assume because of the mutation at some region of the protein can affect the conformation on the epitope where the antibody binds. So, and still we cannot conclude if with this mutation on the opposite, for example, side of epitope of the protein, and we see it affects binding, but we cannot conclude that this is a part of epitope. We just can conclude that it affects binding of antibody. Structure-based prediction became possible only recently. So basically in 2010, about 100 representative structures were available in protein data bank with resolution below 3 angstroms. So the resolution which is suitable for making any judgment about residue contact based on atomic level. And this number unfortunately doesn't grow too much, because in 2012, for example, less than 150 representative structures were in the database, and now the number is about 300, not much than 300. We have two methods based on protein structure, and they work only with protein structure. They do not assume that we know anything about antibodies, which can be questionable assumption, obviously. So we have DiscoTope method and ElliPro. So let's look at DiscoTope first. So we host two versions of DiscoTope. The first was published in 2006, and the second in 2012. And the method is based on a linear combination of the normalized values on each residue using hydrophilicity scale, amino acid statistics, the number of contacts with antibodies, because the method was trained on protein antibody complexes from protein data bank, and also surface area of relative solvent stability of each residue. And the method requires knowing protein structure, either from solved by X-ray from protein data bank or modeled using some modeling software. And I will tell you a little bit about modeling later. And the method assigns to each residue a score value, and based on that threshold, one can tell if this residue can be a part of epitope or not. And it was trained on 75 X-ray structures of antibody protein complexes, and DiscoTope also took into account multiple epitopes. So now I need to talk to tell what is multiple epitopes. Here is the structure of lysozyme, where I mapped in different colors the residues which are epitopes of different antibodies. So, and if you look at that, so except this white area which doesn't have antibodies in PPD, all other parts of the protein can be recognized by some antibodies. So we can tell that lysozyme has many epitopes, so multiple epitopes. And if you take into account just like one epitope, for example, in the method you say, okay, I will focus only on my blue epitope, and we'll consider everything as just non-epitope, something what doesn't interact with the antibody. That would be wrong, because there are many other areas which are also epitopes and recognized by different antibodies, like this area and this area. So, and lysozyme is a very well studied protein, and about 200 structures, more than 200 structures in PDB exist for lysozyme and complexes with different antibodies. Question?
Yes. The blue area and the red area, are those medicine structures, because they don't seem like they're the right shape by FAQ?
So the question was if color depicts exactly one epitope. No, not necessarily, because I ran out of different colors. Yes.
So the blue may represent multiple different
Yes,
colors.
yes. So the idea was just to show how the surface is covered. Yeah. So what's different? So, and we provide on the website DiscoTope 1 and DiscoTope 2, and you might be wondering which one to use and what difference between them. So the DiscoTope 2 performs slightly better, but not significantly better based on p-value. So, and it gives AUC value 0.73. Tested on 52 structures. And what's interesting was shown in the paper which published DiscoTope 2, that if we know more than just structure for the protein, if we know the biological units, so if it's complex, if the protein interacts with some other protein, and if you know that transmembrane residues defined for this protein. So then what they did for 13 proteins for which they knew such information, they showed that AUC value was significantly improved up to 0.82 ACM. And this was significant in comparison with DiscoTope 1. So basically we recommend to use DiscoTope 2 because it's better. And but it's kind of slower. So if you're impatient and want to get your results faster, you can use first DiscoTope 1 and then maybe to use DiscoTope 2. But they have exactly the same output. So let's look at them. So this is a page for DiscoTope. So you should provide PDB ID and its chain which you are interested in, like for protein chain, and it can be several chains, or you can submit a file, which can be a model or file which you can download from PDB or any proprietary file. And then here you can select which version of DiscoTope you are interested in, and okay, so choose file, chain, version, and click submit. So, and for example, for that example, for this particular structure, again, you will see familiar graph. This is DiscoTope score on this y-axis. And the threshold can be changed. And the help for DiscoTope provides some guidance on what different thresholds mean. So I recommend you, if you decide to change threshold, I recommend you to look at the help page and to see what authors of the method recommend for threshold. And again, you can see the same graph as a table. For example, this table, and every residue would have a score, DiscoTope score. And in red, there will be residues which are predicted to be part of discontinuous epitope. And clicking on 3D view, you can will get JMOL loaded, and now you can start playing and to see where each residue is located. So here in yellow, in that representation, you can see predicted epitope residues. But if you want to focus on some specific residue, for example, this one, you click CPK, and then you will see that it will be presented in those CPK representation. So, and again, this is JMOL, which you can play using right mouse and do different kind of manipulation with that inside that tool. So it allows interactively to play with the structure. And coloring the structure and labeling and changing doing some measurements of distances between atoms and residues, etc. Yes, for example, here I selected render, and then I selected hydrogen bonds on, and what I can see all hydrogen bonds in those dashed lines which are in that structure, atomic hydrogen bonds. Okay, so the next method which we have on our website called HDET.
So where is this CNS? How do you know where the number at the graph?
Which residues are epitopes?
For example,
Like
it shows the location on this graph, but
yes.
if we want to translate it to that, the radius, then say the.
Okay, so if you go back, that will be your sequence. It's in sequential order. It just starts from 108. So it's your sequence going on, and those are values assigned to each residue.
So this is just one showing 100.
Yes, so the method gives one value, one score for each amino acid, and so all amino acids which are above threshold are predicted as epitope residues. Okay, so ElliPro method. ElliPro is very simple method, and strangely it performs pretty well. And I recently reviewed the paper where authors considered different methods and did a new benchmark, and ElliPro was one of the best methods. So maybe it already was published, I don't know. But so it's based on very simple ideas. So we didn't invent anything here. We just considered paper published by Jeanette Thornton, where they approximated the protein as an ellipsoid, which is obviously, so, and why I'm telling you that, you need to, when you are dealing with your protein, and you can look at the structure of the protein, if your protein is like cannot globular, is not globular, something like very wiggling, has wiggling structure, then you probably shouldn't use ElliPro because it's not designed for such protein. But if the protein is pretty compact and globular, then ElliPro is a good method to go with. So because it starts with approximating protein as an ellipsoid, and it calculates a so-called residue protrusion index, protrusion from the ellipsoid. So, and more residue protruded outside the ellipsoid, the higher score is assigned. So it's a very simple assumption. And then the method just cluster neighboring residues based on this protrusion index. So the idea is very simple. So, but what's also good for ElliPro, it allows to run modular. So you can submit a sequence, and it will automatically run modular, which is one of the best modeling, structure modeling software available. And then it can proceed with the model to predict epitope. So let's look how it works. For example, I selected from example. So we have two sets of examples. So you can start with sequence, or you can start with structure. So let's look at sequence structure first. So I selected this specific structure, which has structure in PDB. And here you need to select input type, either protein sequence or protein structure. So I selected protein structure. So you can keep all those, so those will be affected if you submit sequence. So here your PDB ID, or you can submit a file again, model, or some proprietary file in PDB format. And those scores, you can look at the help in the paper, which also discuss how scores affect the performance of the method. So if you click submit, ElliPro would predict linear epitopes and also discontinuous epitopes. And for each epitope, they will be given score, like average score for each epitope. So, and also clicking on view, you can see the predicted epitopes highlighted on the structure. And again, it's JMOL, which you can render, rotate, and play with. Let's look at what you can do with sequence. So if to select a sequence, so here you should select protein sequence input type and submit the sequence. It can be just either sequence in plain format or FASTA format. It's FASTA format. And then you can select expectation value for search of that specific sequence in protein database. So the sequence will be, the modeler will be looking for homologs in protein database to do the model. And also you can select the maximum number of structural templates to consider. So, and this is the result. So it gives you the blast, what values are from blast, score, E-value, length of alignment, identities, gaps, and clicking on view, you can look at that alignment. So just to see how well submitted sequence is aligned with some proteins from protein data bank, and decide on your own if you want to proceed with further with the specific template. So, okay, for example, you like the alignment, then you select that structure as a template.
Yes. How will you decide that identity or?
Okay, so how you decide? Actually, I will tell you about this later. So the question was how you decide if template is good or not. So I will show some examples later, but now I can tell that you would judge by E-value, also by identities, and there are some rules in the modeling, homology modeling involved. For example, if the identity is below 20, it's probably not suitable, very suitable for the modeling. Also, you need to look at the length of alignment. For example, like here, you saw that the alignment covers practically all your sequence. So, and it will be good template because all your sequence will be modeled. But if some template has a very short alignment, it can be even shorter, it can be 25 radius. Then you probably think, okay, it's not that interesting. If I have a protein like 200 residues, and I have my template of 50, so
this is the percentage of the length, or is the exact number of that alignment length?
Alignment
is 92%.
It includes depth. Alignment length includes depth. So so that means alignment means so you have your sequence and you have the sequence found in protein database. And if when you look at the alignment, you can see, for example, the sequence from protein database covers just like N-terminal of your sequence and just like first quarter of your sequence. And you can think, okay, if I use that as my model and then do prediction, that will mean I will have no information about the rest of my protein. So maybe it's not very good model to proceed. So, and I will touch a little bit later on modeling itself. Yes?
So we have a question for our webcast. Is conformational change will conformational change in prediction. For example, if there are multiple structures with different conformations, one person, which conformation should be for the prediction?
Okay, the question was if the conformation of the protein and change the prediction. For example, for some proteins, there are different structures in PGD obtained in different conditions, and they may have slightly different conformations. And yes, if you use different conformations, the predictions will change because it considers the structure. So, but again, you need to look at how much your conformations change. So, but probably if you have many structures, go with all of them and compare the results. Yes. We have a very good example with RST. We can discuss it on the results session, by the way. Okay, so for example, in this case, I selected that template and I click submit. And again, I will see prediction for linear epitopes and discontinuous epitopes. And in this case, I selected to visualize discontinuous epitope, so in JMOL. So as you can see here, all values start from 0.5 because on the input page, I use the default value of 0.5 score, but you always can cut to have higher cutoff. For example, you can say, I'm interested only in scores above 0.7. Then that means you will have 2 predicted epitopes for discontinuous, and you will have 2 predicted epitopes linear epitopes. So it's up to you how you would like to play with the tool. And in case with RSV, by the way, so in case when you have multiple structures, it's interesting to try different structures and to see what, and if you know exact epitope, you can come up on your own with a threshold, which can be predictable for your specific protein. So because of a limited number of data, we cannot do that for each specific protein, and I will show you on how many structures all those methods will take. But if you have for your protein of interest, like what we did for RSV, we have 4 different structures, and we know epitopes for some structures, we started functional epitopes. We started playing and seeing which cutoff we can use for prediction for another structure. Okay, and another epitope, and again you can see residue scores in the graph and in the table. Okay, so and a little bit about benchmark. So the first benchmark we did for different methods. So we looked at 4 methods based on structure: ElliPro, Epitope, ElliPro, and DiscoTope 1. And we also looked at 2 antibody protein docking methods. And at this time, we had only a set of 42 X-ray structures, independent. And you can, as you can see now, AUC values were not very encouraging, especially for docking methods, by the way. So, and the most recent benchmark was published in 2012, and they used slightly larger number of structures, 52. But again, as you see, we don't have hundreds structures even. So, and they again saw that no method gave values above 0.73. So, but more methods appear in comparison with previous benchmark.
So yes. The antibodies rate is what monoclonal antibodies?
The question was, is the antibodies to be used in structure, in crystal structures, should be monoclonal antibodies, not necessarily. So we didn't look at that. So we just video focused on they all them.
They have to be, otherwise
I never looked at that actually.
Okay.
Okay.
So these are not naturally occurring now.
You can look at in IEDB, just search for structures and to see the monoclonal. For some, you might don't have information. Okay.
There's none there. Monoclonal doesn't mean all the same.
Yeah, there's the
next generation.
Yeah.
They have to be the same protein otherwise.
Yeah, because you need some concentration for the same molecule. Yeah. Okay. Thank you. Yes. So the question was if any method takes into account modifications in progress. No, they don't. So they just use the structure, what's available in the structure. Yeah, if the modification is there, it will be there. So we just look at that. So it's probably not enough data to separate based on modification. Okay, and in conclusion, for structure-based methods, I can tell that docking methods they perform fully in our benchmark, even in the case of bound docking, when the same protein and antibody was taken from the same structure and tried to be docked back. So, again, in general, prediction based on existing methods poor. So maybe Bjorn will show you the method which gives better prediction. Part of the reason for the relatively poor performance is obviously again the quality of the benchmark data. As I already mentioned, that if we can add some more information in the prediction, like to know interfaces of biological assemblies, and if you know count the multiple epitopes and interaction with the membrane, the prediction might be improved. Okay, so now I will show you some examples and some tips for ElliPro specifically. When no structural template is available, for example, you produce the sequence and ElliPro says no template, I cannot predict. So what can you do then in that case? For example, that specific example was given us by a user from the previous workshop. So, okay, so I submitted that sequence, and yeah, what I got no, there is a template, but they are very bad. So the identity is very low, and I said the identity even below 25 is not good to use, but below 20 already no to go. So the identity in those cases was very low. So and the length of the alignment, as you can see, was just very tiny. The protein is like 150, and the alignment is very low. So in that case, we suggest not to proceed with calculation because what you will have in the end won't help you. So use sequence-based tools instead. So another example, so if again, if our tool doesn't give you template by some reason, you can try to find a template outside IEDB. For example, for this specific protein, the length of alignment again was too short, and I decided to look for a better model outside IEDB because here the templates are selected by one program, by modeler. So, and how it specifically does it, so if you use some different approaches, you might get different results. Also, it depends on how often the database is updated. So there is a protein model portal, and this you are not familiar with that model, protein portal, then I will go through that and explain how to use it a little bit. So, for example, here I submitted my sequence, and I got that result. So there are some models. So that's 6 sequences I submitted, and those in different colors. No, they all will be in the same color. Just yellow is the one which is selected. So if you have click here, then it will be selected also here and over there. So, and as you can see, there is no template that would cover the whole protein. So, and in that case, you just need to think what you want to look at. If you are interested in that region of protein, or this protein, or that region of protein. Because no model gives you anything just for everything. And also you can, if you're not satisfied with that, and you can proceed with, okay, so it's show the model, different models. Yeah, you can look at the specific models, clicking on them, and to see that, for example, this model is very old. You see, it was not updated since 2005. That means you can submit it for recalculation. So that's what I wanted to show by this slide, that sometimes you need to recalculate a model. So you click Submit, and it allows you to choose among different servers. And depending on where you're from, academia or industry, you can check the box and proceed, because some are restricted for industry use. So, and unfortunately, you will get results from all those servers independently. So they will send you just email. So it will be a lot of work to go through them and to see which model you want to select. But all models eventually will lead you back to that file where you can see the quality of the model. For example, here they say, if I look at that, they say the quality is very low, so, and I shouldn't expect maybe to proceed. And that's what I got as a model. So you can learn on the help page how to read all those information. Okay, so what you can further, so for example, you're satisfied with the model. You can download it as a CTB file, and now when you download that, you can submit that. So you select protein structure and you submit that file to the ElliPro. Because I choose that file, and now I got my prediction for a linear epitope and discontinuous epitope. Again, we already looked at, we saw only one part of the protein. So that we need to keep in mind that in that case, we need to interpret the result with caution, because we are prediction for only part of the protein. That means that your epitope can be actually inside the protein. So it cannot be real epitope. Okay, and yeah, that's all what I had for historical prediction. So we can go through some details in the break session. Yes? Any questions?